The overall goal of this procedure is to convert fibroblasts into insulin-secreting cells without inducing pluripotency or using retroviral vectors.
This method can help answer key questions in the regenerative medicine field and in particular in the cell therapy of diabetes. The main advantage of this technique is that we can obtain a switch in the phenotype without inducing a stable pluripotent state in the converting cells and without retroviral vectors. Demonstrating the procedure will be Giorgia Pennarossa, a postdoc in our laboratory.
To begin this procedure, add 1.5 milliliters of sterile 0.1% porcine gelatin to the 35 mm Petri dishes and allow coating for 2 hours at room temperature. Next, wash the human skin biopsies with PBS supplemented with 2% antibiotic antimycotic solution. Afterward, place the biopsies in a 100 mm Petri dish and cut them into approximately 2 cubic millimeter fragments with sterile scalpels. Then, place 5 to 6 skin fragments into the pre-coated 35 mm Petri dish. Add a droplet of fibroblast medium over each fragment and culture them at 37 degrees Celsius in 5% carbon dioxide. After 6 days of incubation, discard the tissue fragments carefully. The fibroblasts should start to grow out of the tissue fragments and begin to form a cell monolayer. In this procedure, culture the fibroblasts at 37 degrees Celsius.